typically we see the live, what we call the live larvae, which will
Yep.
be the healthy ones. You can also see when they are, and that is not very common, but you can see when they are in like a pupal stage. Sometimes you see adults when you have a second generation, which is something we don't want. You can also see when they're parasitites. You can also see other stuff that is there that is it's not bees, right? So predators, parasitoids, and you can tell the difference. Sometimes when the image is kind of blurry because of a pathogen, for example, you can see the larva, the contrasting colors are not quite clear. So what we do is open them up and just count every single cell, and then we come out with the numbers and percentages.
From the Oregon State University Extension Service, this is PolliNation, a podcast that tells the stories of researchers, land managers, and people like you who are making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Associate Professor of Pollinator Health in the Department of Horticulture. Joining us today is Dr. Armando Falcon-Brindis, an assistant professor and extension specialist in entomology at the University of Idaho's Parma Research and Extension Center. His work focuses on practical field management for the region's diverse specialty crops. As the director of the Parma Cocoon Testing Center, he also provides the diagnostic data that allows producers of alfalfa seed to make critical decisions about their leaf-cutting bee stocks. Beyond the center, he's launching a new program aimed at integrated pest management and pollinator management that promises to modernize how we support seed production in the region. So without further ado, let's travel all the way over to Idaho's Treasure Valley this week on PolliNation. Well, it's my great pleasure to have Dr. Falcon-Brindis join us today on PolliNation. Welcome to PolliNation.
Thank you for having me.
Now, you're a relatively new extension entomologist at the University of Idaho at the Parma Research and Extension Center. I think we met maybe a year ago. It's not that long ago. And you have, what as a, extension entomologist specialist, you have a broad range of responsibilities in addition to the topic we'll be talking about today, the health of alfalfa leaf-cutting bees. Can you describe your position? What kind of problems have you been hired to solve?
Yeah, actually, we met probably over a year ago. I started in January of 2024, and I am a research and extension specialist. So I'm establishing a new entomology program. Basically, my main focus is in applied ecology, applied insect ecology, and I also oversee the Parma Cocoon Testing Lab. My split is 50% research and 50% extension. So it is a very balanced duty here in my position, and I'm really happy to be here, serving this community. One of the main or the core problems, issues in this area that I'm looking at are related to pests and related to specific crops. As you know, this area is very rich in diverse in terms of commodities and crops. So I can always do a number of them, right? But I am happily, starting working with uh, alfalfa, hops, potatoes, mint, dry beans, and the number of issues will be it's a broad range going from, multiple pests and insecticide management, maintaining at the same time, maintaining the pollinators, maintaining the beneficials. So I am trying to, connect the dots there with people and educate them.
I mean, I'm not sure if people quite realize, if you haven't been through southern Idaho, the diversity of crops, which I imagine brings a diversity of strange and wonderful insect beneficials and pests. It's a very
complex system you're kind of situated within. And I guess, you know, there's a lot of crops there that are bee pollinated, and many of them are honeybee pollinated, but not alfalfa seed. So leaf cutting bees do the bulk of the work for pollinating alfalfa. Why are alfalfa leaf cutting bees so effective at pollinating alfalfa, and why can't honeybees do the job?
Yeah, yeah, that was something that probably caused excitement when people realized how effective they were despite their size compared to the honeybee. They're smaller, not quite half of it, but they're smaller in their behavior is critical here. So even though leafcutter bees are not social in a way of having a queen, and drones and workers in producing honey, not at all. Their foraging behavior, the way they visit and the touch the flower and collect the pollen. So that's how they're actually being so efficient at it. Has been said that one leafcutter bee it will be the equivalent of like pollinating work of 20 honeybees. I don't know how realistic is that, but they're very, very efficient. And when you hear about the numbers they put out, you'll figure that.
And I remember hearing this, that honeybees love alfalfa, that they love to visit an alfalfa plant, but they just simply don't pollinate it.
Exactly. Yeah, they can't quite you know reach the well, honeybees typically cheat and they're very smart. And
Yeah.
other bees do the same. They could pierce, the flower and they just reach the nectar, or go from the side. They don't quite like the way the anatomy of the alfalfa flower. So that's why they're not gonna do as quiet, or they're not gonna be as efficient as
Yeah.
leaf cutting bees.
I always love everything in the legume family for bees because it's, you know, the plants seem to really, there's the snap where the anthers and stigma sort of snap and hit various parts. And we have, you know, red clover, which we have in both areas. You know, those bumblebees, when they're pushing their head into the floret, they get hit on the bottom with a column. And I always understood that honeybees just don't, the conventional wisdom is that honeybees don't like to get hit. And so they just
Correct.
get the nectar out the side and don't do the pollinating. So
Yeah,
I mean, it's interesting because honeybees seem to serve so many purposes, but here's one that they don't.
absolutely.
Okay, so I imagine everybody kind of knows, you know, you can test these cocoons. Well, I guess people might not know that these bees are the stage that the bees right now currently across, you know, leaf cutting bees are in a cocoon stage right now in a prepupal stage inside. So they're in these little, you know, they're in a cocoon that's kind of covered with these leaf discs. Yeah, and the cocoon testing center is testing these cocoons. What are you looking for? What's, you know, walk us through the process. Like maybe describe what these cocoons are, and then, you know, what's the cocoon testing center doing?
Oh yeah, absolutely. Well, first of all, I want to highlight this center was built or opened back in, I think it was in the winter of 1994. It's been here for a while. And it was mainly serving, or it has been mainly serving, neighboring states. Besides Idaho, we will include, they were receiving samples from Washington, Oregon, even Nevada. And it was they were serving approximately 50 different growers in this region, which is something that it's kind of sad to see because the alfalfa production, across the nation has been declining over the last decades, and uh, and now we get not even half of that from growers. What do we look at? What do we do here? So and I think you said it right. We want to see whether their bees are healthy or not. That will be the simplest way to see this. But we collect information from different parameters. So Everything is measured in a way that everybody can understand that it's simple and easy. It's going to be in percentages. So out of the sample that you bring or the farmer brings, we will tell him, okay, this is the percentage of live larvae. Okay, that means healthy. That's expected to be healthy individuals.
Mhm.
And then we also give him percentages of pests. Okay, and we'll talk about later. We tell them the percentage of just pollen, like empty cells, dead larvae, machine damage, because that is what that happens when they are collecting the cocoons. Um, and then we can also provide a sex ratio test, which means we can test, we let them hatch pretty much, and then we tell them what is the proportion of male and female. So
Okay.
that's what we do.
All right. I know a lot of this is that there's the hatching, which is just, counting and they're, males are easy to see from females, but a lot of these things are, still in the cell. How do you look inside the cell?
Yeah, and that brings to the actual core of what we do here. So the backbone of this lab is X-ray. So through with that method, we're able to look through just like a regular X-ray, right? When you're hurt and you go check with the doctor, you see your bone. So here we see through each individual cell or cocoon, and then we do the counts. So
Huh.
that is in essence what happens.
Okay, so you can see a couple things that you said previously. You said you can see if it died early. You said that it would look like the pollen hadn't been eaten.
You are correct. Yes. And that is it's one of the biggest questions, among specialists and researchers. Like, we are not really sure. There are different hypotheses and, potential explanations for that. But yeah, we can see just the pollen balls there with nothing, apparently like a failed individual or an egg that probably was not laid, or it was either, the bee mom that
Yeah.
didn't do that, or it was an egg that didn't hatch or failed, or
Okay.
the larva died early. So
Okay, so you can have that happen, and then let's say it eats it all and, spins the cocoon. What do you see? What are some other things that might happen to it and that you would see through the X-ray?
yeah. So and then we can see typically we see what we call the live larvae, which will
Yeah.
be the healthy ones. You can also see when they are, and that is not very common, but you can see when they are in like a pupal stage. Sometimes you see adults when you have a second generation, which is something we don't want. You can also see when they're parasitites. You can also see other stuff that is there that is it's not bees, right? So predators, parasitoids, and you can tell the difference. Sometimes when the image is kind of blurry because of a pathogen, for example, you can see the larva, the contrasting colors are not quite clear. So what we do is open them up and just corroborate that we
Oh.
count them. So we basically count every single cell, and then we come out with the numbers and percentages.
Okay, so by using this X-ray, you can peer in and see things like, did it die? Is it alive? Is it a cell full of little parasitic wasp larvae? Is there some other disease in there? And then you can go open them up and look more closely if something's not resolved. So then the grower gets back. I guess they've got their cocoons, they're about to incubate them for the season. They'll know, oh, I might have to be careful this year with you have to do some kind of procedure to clean something up because I'm expecting a problem.
That is very true. And it's I think it's a critical tool for them, that decision-making tool, because they know whether their bees are, in good shape or in decent shape to be, ready for pollinate your crop, or you need to get new bees or get rid of your bees. You have to make a decision because there's a number, there's a recommended rate, that you want to put out and make sure you're going to get a pollinated crop. You got to get a yield. So this is when we talk about the number of cocoons per gallon. So we're getting a little bit more technical here, but they typically put between 3 to 4 or even 5 gallons per acre, right? And each gallon, it's considered
A gallon of
10,000.
cocoons. I imagine it's like,
Yes,
okay, all right.
it's 10,000 cells or
Okay.
cocoons will make 1 gallon. So they put a lot out there, and that is per acre. So they will put 3 to 4, let's say 3 to 4 gallons per acre. So that will make between 3,000, 30,000, and 40,000 cocoons. So you make sure you're gonna be pollinating 1 acre of alfalfa.
Okay. So you need a lot of them to come out and do their job. And I imagine, oh, the other thing you mentioned was the sex ratio. I suppose the females are collecting pollen. So if you have really a sex ratio that's skewed heavily towards males, that might, I guess you might want to put more bees out or something to compensate.
Absolutely. They desired, or under normal conditions, we typically see a female male ratio of 1 to 1.5 or 1 to 2. So that means it usually have less females naturally happening. So you want the females, you want the females to be ready.
I was thinking about you and I've had this conversation before. Some of these problems can be averted, and I know there's challenges now because some of the methods are no longer available. Like those tiny little wasps during incubation. If you have a lot of them, then they can I guess they emerge and they just go and they start to parasitize. They can really during the incubation phase, the period before I guess they go out.
Yes.
People may not understand the incubation, but I guess there's a period when just before they go out that they could real serious pest damages could accelerate. And I guess knowing that you have a problem can help.
Yep. Yep. When you are reusing your bees, that means that you have a return. So you put your bees out in the summer, typically around the 10th of June, and then they are working, they're pollinating your crop. They hopefully come back to you, right? And then that's your return, that's your yield. Within that process is when they are going to collect, probably some pathogens, or depending on multiple factors, they will collect pathogens. Parasites will visit those bee boards out in the fields, and then you're going to, they always have this populations build up over the time. So you want to keep an eye on that. And then once the season is over, they take their bees back into this cold storage. That's what you're talking about, this process that it's going to be critical, because a lot of parasites will come right before you're ready to put the bees out, but you want to clean them. So you have this, like pest strips, and then you hopefully can kill most of those. But also in the field, they have some traps for beetles and like for sapigid wasps. They could also do that to keep them clean.
Last fall, I cleaned my mason bees and had the same problems. I could see, I had chalk brood and I had, do you ever, is the Cocoon Center ever dealing with mason bees? Has that become an area of interest?
We haven't, but there is an upcoming collaboration with professor from Washington State University, and we will start looking at mason bees using X-rays.
Fantastic. I imagine that information could be really useful.
Yeah.
Okay, so you're a producer, and you have all these bees in storage. How do you get a representative sample out? I mean, there's lots, I've seen them before. There are these, bins and bins and bins full of bees. Or sometimes I guess they can also be not, there's, I guess, loose cell, and then there's, bees that are wintering in their nesting blocks. which I imagine fill up a warehouse. How do they take a representative sample and what kind of information, like what do they get back?
Yeah, yeah. So one typically farmers will have, it's either loose cell, like you said, when they purchase their bees from Canada, or they punch them out. They have their loose cell in big boxes in the storage. They can also have their polystyrene boards or wood boards. So when they have the boards with full of cocoons, they need to punch them out. So there's different approaches and methodologies for that. But let's start with the loose cell. So when they have a loose cell, they need to take a handful, right? And then typically go with three different depths in each container.
Hmm.
So they want to mix them up and bring 100 grams of that sample, which is roughly 3.5 ounces or like 2 cups. So that will
Okay.
make a sample. That's what we recommend when you have loose cell. When you have boards, you need to punch them out, right? So for the polystyrene boards, you want to use probably an Allen wrench. You can just take them out, just punch them out, literally, with
Hmm.
that. Um, we have to know this information because that tells a lot to the lab. And then for the wood boards, you want to use they typically use a larval probe, which is something you can purchase, from suppliers,
I think I have
wood
one.
boards.
I have one. It's like a little screw on it.
Yes,
Yeah,
exactly. It's a quartz screw. Yeah, that's right. And then it will have the diameter and depth of the holes, right?
yeah,
And then you basically take them out. It's the corkscrew. It's glued inside a pen case
yeah,
sometimes, I guess. But once they bring those samples in with
yeah.
that information, they fill out a form, which is out in a flyer. It's online. We test the bees. And well, as we mentioned before, we tested live, dead, parasites, predators, and damage and pollen balls. So they are going to get a report with that, what we call live count, with all these details. And if they also requested another test, which is a sex ratio, they will also get another report for that.
Okay. All right. It's a simple, a great system. You can get all of this information back and you don't have to takes the guesswork out. And if you have a problem or if you have a skewed sex ratio, you can make corrections before the field season. This is great.
Yes.
Okay. I know you are just kind of building the program. You've mentioned this a couple times during the interview, and you have this is one of your many responsibilities. Tell us about some of the things that you kind of are working on in the short term and maybe in the long term that will help Idaho agriculture, but also bees.
Yeah, absolutely. So like I mentioned, I have extension and research responsibilities. In almost all of my projects will have both, I'll be targeting both objectives. So I'm going to call one of my passion projects is the establishment of a new pollinator garden. But it's not just another pollinator garden in the region, which we have many. It's a garden, it's a demo garden, but we're also testing different flower mixes, mulching. We're, showing different plants, native and no-natives. We're teaching people so many things. And the special thing about this garden is that I am connecting or using different supplies from commodity groups, from growers, say the telephone poles that they use for hops in hop yards, and then putting some native mint and putting some alfalfa plants and using some of the byproducts from the alfalfa industry, actually. And then everything it's put in a way here in the garden that people can connect with Idaho Ag. The other things that we are doing are, training sessions and workshops to other educators, but you know, out in the field. we're getting tired of being, just looking at our computers and webinars and more webinars, but we want to be out there, and we're doing like summer sessions, summer trainings for IPM, or integrated pest management. So we're looking at different things from, planting, strategies to pest control and things like that. You also incorporating new technologies. So I didn't say it, but this cocoon testing lab it's an example of it. I brought a scanner that actually allows me to have digital X-ray images. So in the past, we did the regular X-ray, procedure, and it takes hours, of course. You have to go and use different chemicals, and develop the film. So now it's a matter of literally seconds for us to have an X-ray and then just take that to a scanner, and then we have the digital image. That brings a lot of advantage as well, and it's changing the way we do this. It is increasing the accuracy of results because we can zoom in with a high resolution image. We're also looking at, down the road, expanding, our research to other crops and other systems, especially incorporating technologies with the Internet of Things and wireless sensors and things like that, and remote imaging, in relating everything with pest management. I'm also working on things related to regenerative agriculture and see how those practices change or encourage the population of beneficial insects. But yeah, so that is pretty much my wheelhouse right now.
Pretty much. That's a lot of things. And I love the blend of both, getting tools directly into people and showing them about it, the demonstration, but also kind of exploring this whole new realm of precision agriculture, because clearly, it's expanding rapidly, and, making sure that your growers, are ahead of the curve and can continue to lead. I was thinking about the pollinator garden as well. It's just there's so many good seed producers in the Treasure Valley. Everything from like native seeds to exotic flower seeds to crop seeds. There's a lot of different plants that are really well suited to growing there, and they're produced right there. This seems like such a great opportunity to demonstrate.
Yeah, absolutely. One of the things that blows people's minds, I want to say, is that the fact that this garden is in the middle of ag farm, just like that, just farmlands around it, and we put it out last year, and we saw hummingbirds coming in, bumblebees, multiple bee species, and wasp and flies and butterflies. It was like insane. For us, it was very, very exciting to see how a small island-like environment for them, they can change a lot of things. So it is something really insightful for us to show to people. And when we see that, again, that excitement from volunteers, from master gardeners, from stakeholders, even people from other countries that are touring around this area and come to Parma. I talk about the program and I show them the garden, and they're like, "This is amazing." So we also engage with, the local schools, and then we have kids coming out. So it's a good classroom out there.
Fantastic. Well, what an exciting program. Also, I'm looking forward to working with you more closely, and just really giving us this snapshot of, a little part of the pollination world that I think most people don't know happens. There's probably a handful of people who think about cocoon testing, and now a lot more people know about it. I really thank you for taking the time to answer questions about it.
Thank you so much, Anthony. Thank you, everyone, for listening, and I hope you like it.
Thank you so much for listening. Show notes with links from each episode are available at the website pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's a form at the website where you can pop in and say hello and give me feedback. If you want to support the show, remember to leave a rating on iTunes, Spotify, or whatever podcast mothership you use. And finally, if you have the means and you want to help support my lab's effort to document bee biodiversity in Oregon, visit OregonBeeAtlas.org and follow down to the donate button where you can make a tax-deductible donation to the Jerry and Judith Paul Native Pollinator Endowment. Every little bit helps. See you next episode.